Hi friends, welcome to my channel, that is BioTutor. Today I have come some important points of photosynthesis, that is photosystem 1 and photosystem 2. Clear? And quantosomes also. So first of all, what are quantosomes? Quantosomes are the collections of some pigmental granules which will be activated by the photon rays, basically the blue and red spectrum. And they will form one unit that is known as photosynthetic units, and here some complex is involved. Okay, and that complex is known as light harvesting complex or antenna complex. Clear? And in case of light harvesting complex, here some accessory pigments and one reaction center are involved. Reaction center will be formed by only one kind of pigment, that is chlorophyll A. It will play a great role for forming that reaction center. And accessory pigments consist of two kinds of molecules. Some are known as antenna molecules and some are known as core molecules. Okay, the outermost molecules which are present almost in outer part to the core molecules, they are known as antenna molecules. Antenna molecules include chlorophyll A, chlorophyll B, carotene and xanthophyll. Clear? And core molecules, they are only chlorophyll A and B are involved, and reaction center, they are only chlorophyll A will be involved. Clear? So accessory pigments are of two types: antenna molecules, they will be present at the periphery, and some core molecules, they will be considered chlorophyll A and B will form core molecules, and antenna molecules, here chlorophyll A, chlorophyll B, carotene, xanthophyll are involved. Clear? So while the sun rays, basically the photon rays will be falling on these accessory pigments, they will be energized and they will pass this energy to the reaction center, that is chlorophyll A. And this chlorophyll A will be energized, will be activated by the photon rays, and this wavelength ranges between 680 to 700 nm. And while they will be activated, they will release two electrons, and these electrons will be accepted by the primary acceptor molecules. Clear? And it will be happening in cell in normal condition. But in experimental condition, if we separate this reaction center in some other test tube or something, it will be directly activated by this photon rays, because there will be no accessory pigments, because we have already isolated this one. And it will be activated and it will release two electrons, and at that time there will be no primary acceptor for taking these electrons. So what will be happening? These electrons again will be coming back to this reaction center, and while they will be coming back, at that time they will release some wavelengths in the form of energy. Okay, and this energy will be producing one color that is known as fluorescent, but it will be done in experimental condition. Clear? So now I am discussing what are photosystem 1 and photosystem 2. In case of photosystem 1 and photosystem 2, they are also the same thing. This reaction center will be formed by chlorophyll A. Okay, but in case of PS1, here chlorophyll A will be activated by the wavelength of sunlight, that is 700 nm. But in case of PS2, here it will be activated by the wavelength that is 680 nm. That is the greatest difference in between the PS1 and PS2. Okay, and PS1 will be happening in the granums or thylakoids and stroma also, in chloroplast. Clear? And here, and PS2 will be happening in grana also. Clear? And here some molecules are also present, some two types of vitamin, two molecules of vitamin K, and some iron complex, iron protein complex. So many things will be present there, carotenoid molecules, so many things there. Chlorophyll A will be there. Okay, and in here, so first of all it will be activated by the sunlight of photon rays, and that wavelength will be 700 nm, and they will release also some electrons, and it will be accepted by some primary acceptor molecules. And in PS2 it will be happening in granum, and here there are also some carotenoid molecules will be there, chlorophyll will be there, but the amount of carotenoid is more active and amount is higher than the PS1. Okay, and in case of PS1 their chlorophyll will be higher than others, and here it will be activated by the wavelength that is 680 nm. That is the greatest difference. Okay, and another one I want to discuss, that is PAR, that means photosynthetic active radiation. I told you that the PS1 and PS2, here the wavelengths in ranges between 680 nm to 700 nm. So PAR means photosynthetic active, that means in between that 680 nm to 700 nm rate of photosynthesis is higher, that it will be activated highly. That is the photosynthetic active radiation. Clear, students? So tomorrow I will again come with some new part, that is cyclic and non-cyclic phosphorylation.